All right, thank you very much. It's a pleasure to be here. Would be much better though if my PhD student Hadir could be here, but she didn't manage to get her visa on time, so you have to make do with me. But it's her work that I'm presenting, of course. And we're going to talk about an approach to model protein-protein interactions. You're all familiar with these beautiful molecular machines, and you know how much work goes into the painstaking elucidation of these structures and their function. But still, we don't know a lot about the majority of the interactome. You could guess that about 80% of these PPIs are still not known. And we set out to chart, again, the uncharted parts, in this case, of the PPIs, and ideally also to make high quality predictions of the structures that are behind that. So when we're talking to collaborators, they often come with a question like, does protein A and protein B form a complex? Do they interact? And of course, if we tell them, yeah, we think they might interact, we have a follow-up question, well, what's the structure going to be like? So what we try to address in this work is answering questions like, do they interact? Well, if they don't interact, then we don't care about that complex, of course, there's no PPI. But if they interact, we would like to build a model by extracting restraints for restraint-based modeling that we could then use to build the proper structure of this binary complex. And of course, if you can do it for binary, then there are ways to go beyond mere binary interactions. But often we can't really answer this question, whether they interact. So we end up with, we're not sure. And then we would also be able to come up with a reasonable way to determine whether they actually interact. And if they interact, we try to predict the interaction. Otherwise, again, we don't care about that. And we will touch on these 3 points here in this presentation. How do we get to restraints that we can use for restraint-based structural modeling, and how can we predict these interactions on a large scale? And by large scale, we mean usually genome-wide, proteome-wide models. So there's a plethora of different ways to assess the interactome, different ways to construct hairballs that nobody cares about. And the number of different methods is almost as large as the potential interactions. I will not go into all of these methods, but I want to introduce very quickly 2 methods that we care about. And that's cross-linking mass spectrometry, or XLMS, and evolutionary couplings. Now, why those 2? Well, when we set out with this study, we thought they're nicely complementary. Cross-linking mass spectrometry is an experimental method where in vivo or in vitro, you can chemically cross-link proteins. And if they interact, then basically your cross-linker sticks to one protein and sticks to the other end. And by doing some bioinformatics, you can infer the sequence of the attachment points of these cross-links. So you get a mass spectrum, you search that, and magically you end up with information which point of protein B is connected to which point of protein A if they actually interact. The second method is probably known to most of you, that's evolutionary couplings, where we look at both interaction partners on the evolutionary scale, and we try to assess which position in this multiple sequence alignment interacts or co-evolves, to be precise, with a site on the other protein. And again, we learn from that these 2 residues in these 2 proteins should interact. Now, why these 2 methods? Because they're nicely complementary on several levels. So first of all, cross-linking mass spectrometry is a method that can be applied on a large scale experimentally. You can do that on whole proteomes, and I'll show you a study later on. Evolutionary couplings can be applied on large scale, but are a computational method. And many of us as bioinformaticians have been facing that issue. If you tell them, well, I predicted this structure, then that always leads to some sort of skepticism. Whereas if you tell them, look, there's this experimental data that's consistent with my model, then that's much more favorable. So we thought, let's combine experimental evidence and computational evidence. But there are also other advantages. So the cross-linkers are often long-range interactions. Depending on cross-linker you use, you get distance information that is on the range of 30 angstrom, give or take few, whereas these evolutionary couplings are very local. Also, these cross-linkers can only access the surface of the protein set outside the interface. So you get information on distance constraints on parts of the two proteins that are not in the interface, whereas the ECs give you the information on the interface. So they're nicely complementary. And what I'm presenting here today is a pipeline that we call XLEC, for obvious reasons, integrated approach to combine cross-linking data and evolutionary couplings. And we do two things. We predict some sort of likelihood of interaction for the two proteins, and we also build high-quality structural models of binary complexes. And what we need as input are, of course, the protein sequences. We need them to build these evolutionary couplings. If you have it, cross-linking data and the monomer structures. And what we get out of that is then a structural model in one case, or the interaction probabilities. So how does that work? I give you a rough overview. We start on one hand with cross-linking mass spectrometry. We don't really go into the experimental details, but it's basically a well-established method. You incubate the protein mixture with a cross-linker that is bioreactive. You digest, you get a bunch of cross-links, and through the mass spectra, you can really infer which position of the protein interacts with the position on the other protein. The problem is there are false positives and there is a low sensitivity, so the data tends to be reasonably sparse. On the other hand, we have evolutionary couplings. In this case, we take the sequence of the two proteins, compute deep multiple sequence alignments for both of them, and then we concatenate them in a smart way. I'll give you some idea of how that works in a bit. That gives us concatenated sequences. We can then look for evolutionary couplings between roughly columns in these multiple sequence alignments that cover protein A and protein B. And again, that gives us distance information that this point, this residue in protein A should be close to another residue in protein B. And what we're doing is we are using this information, these distance constraints that we get to do restraint-based docking, which will hopefully give us then a decent complex structure that is supported by both computational and experimental evidence. So how can we extract these crosslinks? So crosslink data, we have tools and pipelines for that. I'm not going to go into the detail. In this case, we really took the information, and the information roughly looks like this. You have two proteins, their protein IDs, and then you have the peptides that were identified. And you know that a crosslinker, for example, is attached only to lysine residues. You know the positions where that is. So basically, you have to map these peptides back to the protein structure, and then you know that certain residue IDs are in a maximum distance of, say, 30 angstrom. So you can turn these cross-linked peptide pairs into restraint files for docking by mapping them back onto the surface. The second step is then computing evolutionary couplings. That is an established method. You can read that up in the papers that we co-authored with a couple of people, including Debbie Marx's lab in 2014, 2011, where we did 2021, where we did that on a large scale. You compute deep multiple sequence alignments using jackhammer. These MSAs are then concatenated by identifying interlocks. We basically need to know which protein sequence here corresponds to which protein sequence there. And through the standard maximum likelihood approach of EC as it is implemented in EV Complex 2.0, we can then get a ranked list of these evolutionary couplings. You can do some filtering, And again, you end up with a bunch of constraints that you can use for docking. So we can put all that together. We did that by taking these restraints, searching for the proper structure. Of course, we need to do some preparation. We need to map them back to the potentially somewhat shifted indices of the residues. Then we're using HADOC to do a docking run and some analysis scripts. We do that first in parallel. So we do independent docking runs for crosslink restraints and then for evolutionary coupling restraints. And we use that to do sanity check because we have false positives in these sets. So we filter them out based on the docking run and then do an integrated docking run again that gives us a final set of potential candidates. So in order to test whether that really works, we went out and looked, is there a data set that is amenable to this approach? So we would like to do that on a large scale, but probably a whole mammalian proteome would have been a bit too much. So we decided to go for a subproteome, mouse mitochondria, where we had two very nice published studies by Liu et al. And Schweb et al. We could simply take these constraints. They have been constructed to or processed to false discovery rate of 2 to 5%. And we simply could then merge these constraints. They also have comparable length in the crosslinkers. One is a gas phase cleavable crosslinker, the other is a protein interaction reporter. And they were all from mouse mitochondria. So what did we do there? We took the cross-linking data, and it turns out that if you merge them, you end up with roughly 2,500 cross-linked peptides. They map to roughly 300 unique proteins and 770 unique protein pairs. We split that. We took what we call a benchmark set that we can use because we have a known 3D structure of the complexes, so that helps us to validate the approach. And then we have a prediction set. These are basically proteins where we have the monomer structures, but where we do not know what the interacting complex really looks like. We then compute the evolutionary couplings for these protein pairs. We run the docking approach, and then we did some assessments of the quality. So what did we do? The first thing we do is we applied the metrics from the Capri competition, so the fraction of native constant, ligand RMSD, which is basically the backbone RMSD of the second interaction partner, interface RMSD, and the DocQ score, which is a quality metric for the docking result. And then we also built a logistic regression model that would help us in the setting to actually predict, based on what we see, whether this interaction is going to be true. So whether these two proteins really interact. And this is a very simple model that includes sequence-based features like the quality that went into the number of the crosslinks, or the distribution of the evolutionary couplings, but also docking-based features. And we also are going to do comparison in a bit with AlphaFold. And again, we build a model there. The idea of this model is then to give us a score that tells us, yes, this pair really should be interacting. So let's look at the comparison first. So the DocQ score, there is a threshold that is often used that is acceptable quality. Without going into the details, what you can see is if you use the cross-linking, the protein-protein cross-links only for building the models, then you end up with a very small number. This is the cumulative distribution function. So basically, the numbers of models that you get to a certain threshold is pretty low. ECs are doing a pretty decent job, and if you throw the two together, then you get an increase. You could argue that this is only a slight increase, and that's probably true, but it is not about getting as many of these interactions as possible. It is about the fact that you get confirmed interactions that are consistent with experimental data and with computational data. And of course, you could ask, well, there's also AlphaFold multimer. So can't you use that? Isn't that doing a better job? Well, indeed, if you use AlphaFold multimer on the benchmark data set, you will get a curve like this. So this is obviously what we're doing is much inferior to that, you would think. But of course, these are known protein structures. So of course, AlphaFold multimer can sort of learn that thing and is reproducing that. So we were wondering, is that really true? Or are we sort of falsely accusing AlphaFold multimer of learning what it knows and then reproducing that? So we basically looked at the data set where we're doing novel predictions. And what you can look at is the DocQ score for these models. And AlphaFold multimer generates a predicted TM score. And we were basically looking at what DocQ score that we know that is score of 0.23 is often acceptable quality. That would then translate to a PTM above 0.5, and you can see that this goes up. So now we're looking at this score, at this PTM score for our benchmark set. Again, these are known PDB complexes, and for the novel predictions. And not surprisingly, we see quite a difference in these distributions. So it is why we cannot exclude that, but it is sort of likely that indeed these AlphaFold predictions are of a lower quality. And the other issue is that you don't know whether the prediction is true. So we try to integrate then the AlphaFold based predictions as well into our logistic regressions. And since we're already a bit short, I will not go into the details there, but of course, integrating this additional line of evidence that you get out of AlphaFold also increases the performance there. So if we want to try to predict the interaction, then we can do that with an AUC of about 0.84. Now, a few results, what does it look like? So first of all, our initial assumption was quite accurate. So this here is a model, and you see for the benchmark data set, we can predict these binary complexes with a ligand RMSD of between 1 and 2 angstrom, which is not too shabby. You can see that there are these purple lines. I hope you can see that well on the screen. These are the EC constraints, and they're zigzagging in the interface. They're basically sewing that together. And the red ones are the long-range crosslinks that are longer. More interesting are de novo models. So we managed to pull out of this mouse mitochondrial proteome about 500 new interactions. And if you're interested in protein complexes in the mouse mitochondria or mitochondria in general, just let us know. We'd be more than happy to share these models and discuss them with you. They look very, very nice. I think these models, while not all of them are going to be correct, are rather convincing. And we hope that we can discover some new biology in there. So in conclusion, I hope I could show you that XLEC is an integrative approach for cross-linking data and evolutionary couplings to predict high-quality protein complexes that are both highly reliable and also experimentally validated to some degree. We can apply that to large-scale data, to thousands of proteins, and the whole thing is available on our website. Last but not least, that's all the work of Hadir, who is also online, so you can also ask questions in the chat. And I would thank her and my whole group for that. And I would also like to point out that Hadir is about to conclude her PhD. So if you need an excellent postdoc or junior group leader, just let her know. Thank you very much.
So 1-2 questions. So I'll ask while waiting on another question. So how sensitive that method, especially cross-linking for transient interactions, when it's a small interface and they're very quickly dissociate?
That's very hard to judge. Cross-linking can capture some transient interactions. The problem, however, is not transient versus more high affinity interactions. The problem is the sensitivity of cross-linking. So we see many of these peptides we cannot really see in the mass spec. and that is probably more the limiting factor.
Does this apply for all types of proteins, especially when one of the proteins in the pair is a membrane protein?
Yes.
And if one of the proteins is glycosylated or some other modifications are present?
Well, some limitations apply. We haven't tried it on glycosylated proteins, and I think that is in parts not a problem because you can strip them. But if they're fully glycosylated, then the cross-linker will simply not have a place to connect to, and you might have issues getting